Microscopy is one of those things that often sort of slips through, and students get so excited by organelles and cells and yay, we've left molecular biology, that it sort of disappears in the sort of mist of everything else that's more interesting. But the thing about microscopy is that there's some important concepts that you've got to get going, as well as the ability to just look down a microscope. So what this lesson is doing is taking a little bit of step back and focusing on some slightly different elements so that we can pull everything together. So we are looking at magnification and resolution. And then we shall compare light and electron microscopes. Magnification and resolution. Two very different things. Yes, they're sort of working together in tandem, but it's really important that you can sort of split them out and look at them independently and understand exactly what each one is. So when we're talking about magnification, we are talking about how much larger the object is when we're looking at it, so as our image, when compared to its real size. So magnification is how much larger the object appears when compared to real size. So that, of course, is our little formula. Magnification equals image over actual. And that's all there is to it, okay? So when we look at it, how much bigger is it? Resolution is a little bit different, okay? Resolution says, can we see that those 2 points are different? Or is it one big fuzz? So resolution is our ability to distinguish between two separate points. And in this age of digital imagery and digital photos and zooming in, you actually have quite a lot of experience of this before you even get to microscopes. So let's say you take a picture and then you zoom in on it. You are magnifying something in that picture. Resolution is how clear is that picture? When you zoom in, is it still a clear picture, or do we get to a point where it goes fuzzy and now you're not sure where the edge is? Okay, so it's dependent on your pixels and the size of your pixels and the amount of your pixels and all of those things. So nowadays, with our lovely new technology, you can zoom in an awfully long way before things get fuzzy. But if you think back in time a bit to sort of the earlier digital cameras, you reached a point quite quickly where you zoomed in and things were fuzzy. So we've lost the resolution. Okay? Our pixels are not fine enough in that case to give us the detail of the image. So magnification is zooming in, resolution is picking up the details. So if we want to combine them, we would say that we want higher magnification and we would want finer resolution, and this would give us the ability to see more detail. It doesn't actually matter how good our magnification is. If our resolution is not there, then we can zoom in all the way and we can just look at fuzz. Okay, resolution is actually the key to being able to see things, because resolution is our ability to distinguish. Magnification is just how much did we zoom in. Okay, important distinguishing feature between magnification and resolution. So obviously when we then take a look at it, we say, okay, magnification, yeah, okay, right, now I understand. It's just getting it bigger. So resolution is the one that I've really got to get my teeth into, because this is telling me the difference between the light and the electron microscope, what was the point of the electron microscope? It's actually that resolution. So let's talk a little bit then about how resolution works. It's all about wavelengths. So let us have a wave. Don't take this to your physics teacher. But there we have a wave of radiation. Okay? If we put something in there, our wave just keeps going. If we put something else in there, we have now interrupted our wave. This one is not visible. This one is visible. Because resolution is all about that interruption of the wavelength. Okay, so in actual fact, we'd make this one visible and then we wouldn't have the wave continuing afterwards. Okay, so the idea is when we interrupt that wavelength, this is not a very efficient tool to use. Let's see what we can do. Can we not make it wider? There we go. Okay, so we disappear the wavelength. Okay, so basically as soon as we interrupt that wave of radiation, now all of a sudden something is visible. So now let's go back to our normal width, find our green pen. Okay, so resolution is about interrupting the wavelength. That is not our normal width. So interruption creates visibility. Are we happy? So what determines which blob is visible and which blob is not visible? It's all about size of the blobs. Okay? We can say that the limit of resolution is half the wavelength of the radiation used. Okay, so let us get us some wavelengths. Whoa, sorry, something's just gone very funny with mine. Okay, so there we have a nice big wavelength. And then let us have a shorter wavelength. And then let's get going with some blobs. So if we look at a blob of this size, nobody's going to see it. If we look at a blob of this size, we are definitely interrupting both wavelengths. So we would see it on both sources of radiation. If we had a blob of this size, you would see it using the blue radiation. You would not see it with the green radiation. Okay, so we can think of all sorts of different sizes of blobs. Okay, and basically, depending on that wavelength, if we've got a long wavelength or a short wavelength, then we're going to be able to see different sizes of things. So if you want to now turn this into something more technical, our blobs, of course, would be organelles and membranes and all sorts of fun internal structures. Our green one here would be the wavelength for light, and our blue one would be our wavelength for electrons. So what we're saying is, no matter, irrespective of how far we zoom in, we're not considering magnification here. What we're actually considering is resolution, because that is our ability to then see something, to say, ah, there is a blob here, or there's just smoosh. Okay, so when we take that shorter wavelength of the electrons, then we can see things that are much finer detail, much smaller organelles. We can see cell membranes of 7 nanometers. Okay, if we're only using that light microscope, then we've got much bigger organelles, and we can't see the detail within them. Okay, we see a nucleus. Okay, it's a blob. We can't see all the fine details inside the nucleus. We can't see the nuclear envelope itself. So what we're doing then is we're saying, okay, this is the difference between the light and the electron microscope. It's really about this wavelength. And that's why when we talk about resolution in this lesson here, we keep talking about radiation, because it can be light or electrons, depending on what you are working with. So, light microscopes. Their radiation source is light. Magic, hey? So we need to think then about what would be their maximum resolution. Well, remember we said that the limit of resolution is half the wavelength. Okay, so what is the shortest wavelength? Okay, our 400 nanometers is our smallest wavelength for light, for visible light. So that would mean then our best resolution would be half of that, 200 nanometers. So if you are smaller than 200 nanometers, not going to be seen on a light microscope, you're undercover. You're just part of a fuzz. Okay, so we can get to 200 nanometers. If you're closer than 200 nanometers, you're not going to look like two different things. You're just going to look like one thing. If we think then about electron microscopes, our radiation source is, no prizes, electrons. Ooh, if you can spell electrons, it helps. So, electrons, of course, have a very different wavelength to light. Their smallest wavelength is a mere 1 nanometer. So our best resolution is half of that, 0.5 nanometers. So now if we think of something like a cell membrane, 7 nanometers wide, much bigger than a 0.5 nanometers. So this is now why we can start to see all those wonderful details and the internal structures of cells when we're looking with an electron microscope instead of a light microscope. So how do we actually see the thing? That's an important point to consider. It's, of course, your light is visible here. So you look down the microscope and you see the light, okay? When it's an electron microscope, different thing, okay? We can't see electrons. Gosh, life would be quite different if we could. Okay, so we have an electron beam, we project that onto a fluorescent screen, and then we digitally alter that in order to be able to understand and see the image. So here we have a digitally altered image. Okay, and then you've got your transmission electron microscopes, the TEM, and your scanning electron microscopes, your SEM. And each of those are giving you a different kind of image. So a transmission electron micrograph, then you've got the electrons that are actually coming through your specimen, a super, super, super thin specimen. Okay, so it looks much more like a traditional microscope slide. And an SEM, you're actually looking at the surface of something. So you can get the really amazing details of that surface in like a 3D representation of the surface that you're looking at. So different images, different values, different points of use, okay, depending on what you're studying, what you're looking at, that will then affect which method would be the best. But you've got a whole sort of split, of course, between your light and your electron microscopes. So we've covered this idea quite thoroughly now, that when it comes to the major difference between them, it's all about resolution and a resolving power of the microscopes. How fine can you go? How much detail can you see? So there are quite a lot of other differences between light and electron microscopes. So let's have a look at some light versus electron microscope concepts, problems, issues, delights. Why do we even use a light microscope? So we shall have light microscopes on the left, and we shall have electron microscopes on the right. So for a light microscope, you can actually look at living tissue. Okay, so you can actually observe processes. Electron microscope, on the other hand, everything must be deader than dead. Okay, so we can have living tissue. We can observe processes. Whereas an electron microscope, fine detail as we may be able to get, it would have to be dead tissue and therefore no viewing of processes. So on a light microscope, you could watch cell division. Okay, on an electron microscope, not going to happen. On a light microscope, moisture is fine. In fact, we often add moisture. On an electron microscope, not a single solitary drop of moisture. It must be fully dehydrated specimens. Okay? Your specimen, your electron beam, and your screen are all in a vacuum. And therefore, if you have any water in there, you're going to have boiling at a very much lower temperature than you would normally. And of course, boiling water in your specimen will create a massive problem in terms of what you're looking at. So for an electron microscope, we need to have that fully dehydrated. So that's why I say it's deader than dead. Okay, so for a light microscope, we do it ourselves. AS Biology includes making our own microscope slides. So obviously, slide creation is relatively easy and straightforward. Maybe not quite the first time you put a cover slip on, but it does get easier with a bit of practice. So our slide prep is easy and straightforward. With an electron microscope, goodness gracious, not a chance. Okay, slide prep here is complex. It requires training and a lot of practice. Other thing, remember we talked about we have to digitally alter those electron microscope images. Okay, so in a light microscope, you actually see what color it is. You can observe the natural color. This is, of course, if you haven't stained it. If you stained it, then not quite so much of a natural color. But if you don't stain, you can see chloroplasts are green. Okay, with an electron microscope, it doesn't matter if you stain or not stain, you're not going to see green chloroplasts because we're not looking with a visible radiation. We're looking with electrons. So here, we can add digital color. We can color in our chloroplasts so that they look green, but it is false color. Okay, we can color them whatever color we wanted. If we wanted, we could have pink chloroplasts today. Okay, so a light microscope, we're really getting a better idea of what is really going on, what's really there. Electron microscope, we can get that detail, but we don't know what color it is. For all we know, the inside of a cell membrane could be neon pink. But it's unlikely because I do know things about pigment molecules and such like that they can get proper ideas of what colors are. But it's that concept of an electron microscope, when you're looking at a micrograph, it's all false color. It's all something that has been digitally altered by a human, rather than when you're looking down a light microscope and you can actually see what the color is of what you're observing. With a light microscope, we can use special stains for special tissues. Okay? We can stain all the lignin in our xylem bright red and not stain anything else in our specimen. Okay? So we can stain differentially for different tissues. When it comes to an electron microscope, we can stain, but we can only stain with heavy metals. And that is not going to pick up different tissues. We can't do heavy metal staining just for the lignin. So there's a lot more expertise that is involved in also understanding these electron micrographs and really figuring out what's there and what's going on. And our last one, the relatively sort of deciding factor on top of all of this, is that a light microscope is relatively cheap to purchase and service. Okay, schools have collections of light microscopes. You as an individual person could buy a light microscope if you wanted to, and you would be able to buy it and you would be able to service it regularly, and it would all sort of fit in within a normal person's budget, if you budget for that sort of things instead of maybe washing machines. But yes, a light microscope is something that you could save up and buy. An electron microscope, you would be saving for an awfully long time if you as an individual person wanted to buy an electron microscope. These guys are very expensive to purchase, but not just that. You know, a light microscope, once you've got it, you plug it in, you get some slides and you can go for it. An electron microscope, you've got to maintain it. It's also expensive to maintain. And it is expensive to run. So an electron microscope is a real specialist tool. It's not the sort of thing that we have in our school labs. So there you go. Highlight of tables of differences between light microscopes and electron microscopes. Okay, these are differences in addition to that difference in resolution. So let us put that in at the very end. Whoa. Okay, now we're just going a little bit over the top. So, best resolution equals 200 nanometers versus best resolution equals 0.5 nanometers. So it's really important that you understand what resolution is and how it makes that difference in terms of visibility. Magnification is just how much bigger is my image. Resolution is all about what can I see. So light microscopes are fantastic. Electron microscopes also fantastic, but we have different roles for the two different types of microscopes.